Hello, everyone. Welcome back from lunch. I hope you enjoyed it as much as I did. This is okay. Hi, my name's Justin Kinney. I'm an associate professor at Cold Spring Harbor Laboratory. I'm the moderator for this session. So for this session, we have the session is occurring in 3 chunks. It's going until 2:30 p.m. And up next, we have some poster pitches. Is that right?
I'm developing a cell-based assay to functionally annotate every missense variant for the DNA repair gene MUT-YH, which repairs adaxaguanine to adenine mispairs. I adapted an assay known as GFP-off, containing an adaxaguanine to adenine mispair, which, if repaired, results in cells turning on GFP expression. I complemented HAP1 MUT-YH knockout cells with cDNA of MUT-YH wild-type and loss-of-function variants, and transfected these cells with the GFP-off construct containing this adaxaguanine to adenine mispair. I used flow to analyze these cells and found that the nonsense complete MUT-YH loss of function exhibited practically no repair of this construct, and the nearly complete loss of function variant Y151Z repaired at about 5% of wild-type levels. The partial loss of function variant G368D repaired at about 30% of wild-type levels. These data match previous reports and show that this assay can differentiate between different levels of functionality and is promising for its use as a high-throughput assay.
Hello, my name is Ian Hoskins, and I'm a student in the Genic Lab at UT Austin. Current software methods for analyzing multiplexed assays do not easily scale to large genes. We developed a package, SatMut-utils, to simulate and call variants in a scalable manner. By simulation, we conducted the first benchmarking analysis of MAVE variant callers. We further determined mRNA abundance for thousands of variants in an important metabolic enzyme. We found a correlation between codon stability and variant effects on mRNA abundance, emphasizing the regulatory potential of coding variants. The SatMut-utils package enables high-performance analysis for multiplexed assays of variant effect. Please stop by my poster to hear more.
SH3 domains are organized protein domains which interact with unstructured motifs. And one example of this is the SH3 domain of SHO1 and the motif from PBS2 in Saccharomyces cerevisiae. And this interaction is activated by osmotic stress. So what we were able to do is build the DMS library of the SH3 domain of SHO1 and measure the binding affinity of every mutant with PBS2 in osmotic stress. And then we were able to see how these measures of binding affinity compared to other measures like the stability of the SH3 domain and different measures about the amino acid which are modified. Furthermore, we were able to show that our measures did agree with calculated stability changes from Foldex. So if you have time, I'd really like to talk to you. So if you can come and see my poster entitled Exploration of the Sequence Space of an SH3 Domain from Saccharomyces cerevisiae, and I'm David Jordan.
Defects in CPOX cause hereditary coprolpharia, a severe neurological disease, and sequencing is considered to be a first-line diagnostic tool when porphyria is suspected. Unfortunately, most of the missense variants in CPOX have been interpreted as variants of uncertain significance. So we set out to experimentally assay the function of all possible missense variants for CPOX. To do this, we developed a yeast-based functional complementation assay and coupled it with our established framework for exhaustively mapping functional coding variants. This then generated a comprehensive variant effect map for CPOX. We obtained measurements of functional impact for 87% of all possible amino acid substitutions, and the resulting map could recapitulate many known features of CPOX biochemistry. But most importantly, the map captured 58% pathogenic variants. So our CPOX variant effect map provides functional evidence that's proactively and immediately available to assess diagnosis of both known and yet to be observed clinical variants.
Hello, my name is Kai Yue. My thesis topic is improving the interpretation of real variants in the context of the dystrophin glycoprotein complex. We used lentivirals to introduce all possible single nucleotide variants of a protein called FKRP. Our cloning method is fast and cost-effective. FKRP has 4,450 5S and V variants. Our method costs less than $2,000 and only requires 3 days to make the plasmid library and 7 days in total to make the virus library. The variants were introduced as reverse PCR primers and extended into the full-length sequence using a two-way extension strategy. Our final screen results generated functional scores for each of the variants. Our functional scores correlate well with the clinical reports.
We develop reporter cell lines for deep mutational scanning. Development of these reporter cell lines requires the stable introduction of the wild-type protein of interest. This allows us to accomplish a proper reporter gene response. A mutant library of the protein of interest can then be introduced using the highly efficient BXB1 integrase, so that in each cell, one mutant displaces the wild type, resulting in a switch in expression from wild type to mutant. Unwanted random integration can be monitored via a CD20 surface marker. We developed such deep mutational scanning reporter cell lines to determine the effect of mutations in the glucocorticoid receptor on gene activation and gene repression. The system nicely replicates the gene activation profiles of well-characterized glucocorticoid receptor mutants. We are also developing a deep mutational scanning reporter cell line to determine the effect of mutations in p53 on its protein-protein interactions.